we have about an hour for the discussion, but hopefully we have time, to talk about your work. So maybe say about 35 minutes would be an okay.
35. Oh boy. Okay.
Is that too much or too little? I mean, we're flexible.
Okay. All right. So I'm going to talk about several projects which involve modeling approach. We've been working on for a while. It's called subcellular element modeling. It's coarse-grained molecular dynamics models. So I'll try to explain how we calibrate them for different applications. And on the biological side, all three projects are related to studying interactions between cells, some of them stem cells, with extracellular metrics. And for some reason, so that's my favorite cartoon, which kind of is very relevant to mathematical biology because we talk to biologists and people in medical field a lot, and that's not the way to clarify the results. You don't talk about equations to them, right? You show simulations in silico experiments and try to look at kind of testing new biological hypotheses and mechanisms. And of course, modern biology is heavily relying on molecular biology studies and genetic manipulation, and I'll mention both of them. So just want to mention a workshop we co-organized with Christophe Godin, Philippe Meigny, and Roland Merckx a couple of years ago, which was titled Modeling Shape and Size in Biological Development. And that's exactly what I'm going to talk about. So I'll be talking about two examples of regulation of stem cells, one in Drosophila wing during development, another one in the stem, the upper part of a stem of a growing plant. And it consists of stem cells, and they self-regulate in a way that it maintains specific structure. And I'll try to explain the difference between tissue in an insect or animal and in plants. These are different creatures. All right, let's talk with kind of multi-scale nature of development. So basically, people look at subcellular processes like cell division, polarization, and such. And then they look at just few cells interacting. And then they look at the tissue level, and they start looking at mechanical properties of tissue and shapes. And shapes are very important during development, obviously. So we've been studying actual wing of Drosophila. And Drosophila is a very popular, of course, biological model in biology, in developmental biology. So genome is very well studied. And of course, there are some similarities with human genome. So Drosophila is used to study kind of development of cancer in humans, just by looking at specific genes and gene manipulation. So the hope of kind of studying mechanisms of early development, the goal of that is basically to come up with new types of materials. That's one of the goals, right? So maybe certain tissue can be grown in a way with specific properties in mind, kind of by using basic mechanisms of development. So there are hurdles to developing this kind of combined simulation and experimental approaches. So computational cost may be huge, and comparison between experiment and computational modeling results, kind of that's an issue, and people use sensitivity analysis and nowadays machine learning approaches. And we do the same, and I'll try to explain what exactly we do for comparison. But payoffs are huge. One target is synthetic morphogenesis, as I mentioned. That was also one of the goals of having this workshop at Leiden University a couple of years ago. We might have another one. That one was online. So as soon as we kind of come up with a date, I'm inviting everybody to join us at that workshop again. All right, so this is a wing of Drosophila, and that's like completely developed wing. It has certain structure, and this is kind of where the wing is forming. It's called pouch region, and if you look at this region in an embryo, which is not that difficult to do in a Drosophila, it's a very small embryo. You see, we are looking for, from apical side of the wing. You see that some cells get rounded and then they divide. And this has been kind of studied from mathematical modeling point of view. But that kind of point of view doesn't give you a full picture of biological processes. For that, you need to look at this piece of tissue from a side, and I'll talk about this in a second. But basically, cells in the wing are very elongated. And then they divide only when nucleus actually goes to the top. Because you cannot divide when nucleus is sitting somewhere in the middle because there is too much pressure. So nucleus needs space and expansion of at least one part of a domain of a cell to go through mitosis. So quite a few models being developed, finite element approach, vertex, of course, based models, and I'll talk about subcell element model. These are some of the references. So let me just explain what subcell element model is. So different parts of a cell are represented by nodes, collection of nodes connected by springs, linear and non-linear, and some potentials. So as soon as you start talking about potentials, there's like Morse potential and so on. This comes from molecular dynamics, basically. This model's been first developed and been widely used for studying plasma in physics or molecular structure, like of water, for example. So basically, we track motion of each node, and these are Langevin equations describing kind of this motion, and kind of different interactions are represented by different types of potentials. So E sub whatever, there are special potentials, and we have different types of nodes. So for example, there are membrane nodes representing membrane of a cell, and then there are internal nodes, some of them representing nucleus, can be representing cytoskeleton as well. It all depends on level of resolution or coarse graining. So the question of how many nodes to choose to represent specific part, element of a cell, for example, depends on the resolution of the data. And data is usually, it usually comes in imaging data, in the form of imaging data. And of course, resolution gets improving. We use confocal microscopy images and movies. And this development processes, some of them are happening very fast. So that's the general diagram. And we calibrate mechanical properties of cells by finding ranges for kind of spring constants, for example, for describing interaction between nodes or mechanical properties of a membrane. For adhesivity of cells, that's represented using sometimes springs, sometimes potentials. We do the same. We basically look at data kind of from papers where people applied optical tweezers to kind of pull on a cell or kind of deform it in any way. And then we try to reconstruct simulation, kind of getting the same type of deformations of an individual cell. So we're able to do that. And you're looking at kind of distribution of nodes for all these different deformations and mechanical perturbations here. So this is a simulation of a piece of a wing, the pouch. It's an apical view, and cells are growing. First of all, they're rounding, and then they're dividing and adjusting. And of course, the beauty of this approach, we are able to calculate very carefully distribution of stresses. And some of the stresses in tissue been measured in experiments. This simulation kind of was run with 3 boundary conditions, but it's possible to introduce boundary conditions, kind of linking this piece of tissue to the rest of tissue, to the wing. And we were able to use this model to, for example, get polygon class distributions, which are similar to what we obtained using like general laws of biology, developmental biology, Euler laws, above-level laws. The other important feature of this modeling approach, we started simulations with rounded cells, and that's a natural shape of a cell if it's not in a tissue. And then they adjust to each other. The whole system goes to equilibrium, and we get this polygon kind of shapes of cells as a result of initial calibration. And then we get initial distribution, and we kind of classify them using different metrics, and polygonal class is just one of them. And we choose parameters in a way that initial distribution is similar, speaking of kind of classification with experimental pieces of tissue. And we're collaborating very closely with Dr. Zatman's lab, bioengineering at University of Notre Dame. And they have tens of thousands of samples under different conditions. They work with mutants, and they use perturbations of tissue and so on. All right, so recently we actually coupled a mechanical model with a signaling model. And the divisions in the simulation we were looking at were actually tied up to steady state distribution of chemical concentrations. Actually, now we have a dynamical model. That's the difference. So it makes it multi-scale also in time, not just in space. So just want to mention some other groups.
Sorry, did it freeze for everyone?
Yes,
Okay, so let's just pause.
So I think I've shown this simulation before. That's an apical view. And I started talking about coupling a mechanical model representing mechanical properties of cells, cell division, and so on, with a signaling model. And that's our full signaling model, which involves DPP morphogen gradients. And we kind of couple grid for representing kind of signaling model and diffusion of a signal inside of a tissue with the grid for subcellular element, which is non-trivial because subcellular element model is not lattice based. So nodes move anywhere, and the dynamics of them in space is determined by a whole bunch of Langevin equations. We're actually usually solving hundreds of thousands of equations at the same time. So it's coarse-grained molecular dynamics approach. But then when we kind of come up with a specific, say, polygon in this case, after we run mechanical model for a while, we use triangulation to represent chemical concentrations. And based on previous chemical concentrations, we recalculate them in each of these cells. We allow diffusion through membranes. We get new distribution, and this is used for determining division rates of cells and polarization of them in some cases. So this is one of our recent simulations. We're submitting a paper. When we started kind of, again, domain with free boundary. But we kind of prescribe kind of sources of morphogen in the middle, and cells respond to that, and we get pretty close to what's been observed in the center of a growing, developing wing. So let's take a look from the side at this piece of tissue. This is how it looks from the side. So it's much more complicated, and it's curved. So the shape of this wing to us is basically the curvature. And we use curvature profile. So we take certain points along the profile, and we calculate local curvature and sum it up, average out. We use different metrics to approximate distribution of curvature. And we use machine learning approaches to kind of find these distributions based on experimental data. And this is how experimental images look like. And our collaborators, of course, are imaging actin, myosin to integrins. And there is a kind of variety of different interplays between all the different components and, of course, signaling. So first of all, as I mentioned before, cells are kind of elongated. And you see this nuclei fluorescing in blue, and actin is in green. So it's distributed along membranes and somewhere in the middle of a cell. Integrins are on the bottom and on top of this layer. So basically, there are two layers of cells initially in developing pouch of a wing. And then there are layers of ECM on outside, here and here, so magenta color. And they're columnar, the shapes of them. So we'll be looking at like apical surface and basal surface. And what we observed by looking at images and movies, actually, that this nuclei move very actively up and down, of course. Of course, they need to move up for cell to divide, but at the same time, by redistributing this nuclei at different levels all over the tissue, there is like this emerging kind of shape. You see emerging of a shape or curvature of this and structure of this piece of tissue, which is absolutely unique. And if it goes wrong, something goes wrong, that it's going to impact further development, and the wing doesn't develop properly. Okay, so this is a 3D actually reconstruction of these cells, and you can very clearly see where nuclei are, and this is how it looks. So for studying division and kind of rounding of cells, apical view is good enough because this is where the action happens during division, right? So this nuclei go up, they divide there, and then new membrane is formed. But during all these processes, the rest of the cell becomes very, very thin. And we've been studying it recently. So we started looking into the role of actomyosin contraction in different parts of a cell and how it's controlled by signaling. But basically, these are different phases of cell division in the system. So nucleus get pushed up by actomyosin being very actively contracting on the bottom and sometimes on top. And then division happens and new membranes form. And then kind of this part of a cell kind of opens up. Also, this is controlled and actually kind of due to actomyosin dynamics. So I mentioned that we actually got through a lot of imaging data. We used machine learning algorithms to characterize, classify different shapes and structures. And structure for us consists of two types of curvatures on top and bottom, and then positioning of actually nuclei all through the length of the wing, and aspect ratio of cells and the rest of it. So basically, that's the hypothesized mechanism. We kind of thought that interaction with ECM on the bottom, which I mentioned briefly, is important. And we thought that actomyosin contractility on the bottom was important. And after running simulations, we came up with the following predictions, that this nuclei distributions controlled by actomyosin contractility. That process determines the curvature, but it's kind of unstable without interaction with ECM. So ECM, it's kind of every cell in this piece of tissue is attached on the bottom to ECM, and ECM kind of redistributes the stress, and it doesn't allow individual cell kind of to leave the piece of tissue. So we run computational simulations when we removed part of ECM on the bottom, and then the structure changes completely. And of course, after that, they ran this in experiments, and some cells actually leave the piece of tissue completely. So adhesivity with neighboring cells is not that strong, because you're looking at stem cells. They are not very adhesive yet. There is some adhesivity there. Because you saw distribution of integrins, mostly on the bottom. That describes kind of, that's distribution. Integrin describes interaction with ECM. There's some signaling involved. So we also suggested kind of come up with new experiments, like degradation of collagen, ECM on the bottom at different moments in development, and we predicted shapes, and which would result from that. And then we got confirmation, very clear confirmation experiment. So that's the model. And you can see individual nodes moving. So here we don't have division yet. We kind of ran it for a very short period of time, and still we've seen experiments that shapes can change dramatically without division. And here we removed parts of extracellular matrix on the bottom and changed, loosened up actomyosin contractility in certain parts. And we got the shape which we predicted, and they used mechanical perturbation and chemical perturbation. So it can impact actomyosin dynamics by using certain chemicals, and they got this shape there. So
can I ask a quick question?
Sure.
Just why are some nuclei going up and others are going down?
So it depends on actomyosin contractility, like distribution activity. And this is what we see in experiments as well. So if you look on the right-hand side, you see this distribution of nuclei. They're in blue. Some of them are up, some of them are down. They definitely go all the way up before division, but they always kind of oscillate, which is pretty amazing. Mm-hmm.
Thank you.
Sure. So just want to repeat the conclusions we've got in this paper a couple of years ago. So basically, actomyosin contractility dynamics controls positioning of nucleus, and that's what determines shapes. And ECM on its own cannot actually result in change of a shape if you change kind of interaction with ECM. But it plays a role of kind of stabilizer. It's stabilizing the distribution of stresses, kind of averages out, kind of all over the piece of tissue, and this is very important. By the way, I showed you 2 layers of cells. After a certain period of time, the upper layer actually thins out. Actually, they disappear, the cells on the top, and it becomes just one cell deep layer. And then it kind of, from this shape, goes to folding shape. It folds at the ends and then comes together again. And then when you get the right structure for the wing, and we can model that. So we recently started looking at actomyosin dynamics at a micro scale. So we're developing a model for actomyosin contractility. So we'll model individual motors, myosin motors, and we have representations for individual fibers, actin fibers. And again, it's a bunch of nodes connected by springs with some twisting and so on. So at this scale, calibration is an issue, because in images, we can see individual fibers, actin fibers and bundles and so on. But individual motors are just, they're on molecular level, and the way we model them, that's like kinetic theory model. But calibration can be done at a different scale. So these bundles of actin kind of contracting form rings. And these rings can be made visible, actually. So you have the whole bunch of rings, and this is how contraction happens. And we see them underneath, actually, nuclei, as well as on top of it. So cell constantly kind of squeezes and releases the different parts of the cell, moving nucleus and whatever else is inside of a cell. So basically, we have these interactions, and kind of from simplified point of view, myosins kind of control the shifting of individual fiber. So if you put a myosin on top of a fiber, kind of this is actin fiber, not microtubule fiber. So there are famous kind of cartoons when you see kind of a motor walking on top of it. It doesn't happen here. Myosin is bigger than, of course, microtubules, and I mean actin. And they kind of try to shift the fiber. And then, of course, the right thing to look at it is like you need to look at two heads of the motor, like this is the picture C here, connected to two different fibers. So they shift one with respect to the other. And people looked at kind of experiments calibrating this model outside of a cell, so just by creating polymerization process. But we are trying to calibrate it by looking at how much the cell deforms in specific places. And we see, we can evaluate actually density, local density of myosin, instead of looking at molecular level. But we're modeling this at molecular level. Again, coarse-grained. So these are the equations for actin filaments, different forces applied to different nodes, so bending, and the rest of it, and interaction with myosin. So interaction term with myosin, that's like wide open. We're testing different functions for that. All right, how much time do I have, if I have any?
We can keep going. We're a little flexible. Like we usually run around till 12:30, and if anybody has to go, they can. But you're free to talk more. It's just then we can run the sessions.
Okay. So just very briefly. Yeah, I don't know how much people know about plants, and personally, I got into studying plants for years ago. After my group and I, we moved to UCR from Notre Dame, and we're now collaborating with biologists in plant biology. It's a completely different creature. It's like the way stem cells operate. So in an embryo, stem cells kind of divide, and then at a certain point, they create ECM and the rest of it. And then the embryo develops, right? And then it starts growing as a whole, right? So plants keep growing all their life, right? And the term stem cells comes from stem of a plant. This is where stem cells were first noticed, this cells which are capable of differentiating into any kind of cell. So if you look at a plant, and I'm going to talk about a specific plant in a second. So the maintenance of plant tissue structure in time, that's what I'm going to talk now, using the same modeling approach. But basically, stem, it's like central stem of a cell, of a plant. You can go outside, just look for any plant you want. So what do you think the main difference between plants and animals? On behavioral level, right? So I'll tell you. So plants don't have any legs. They cannot run away from a danger, but they're very plastic. If you block, for example, their growth, they're going to outgrow it. They start changing direction of growth, and then they're going to outgrow. And if you cut one branch, it's going to usually regrow, or it's going to start a different branch. So it's a fascinating system. Basically, it's a different form of life from animals and insects and so on. So the agricultural applications are enormous because of global change, whatever people call it. So especially in California. So this is the Arabidopsis thaliana. So it's as popular in plant biology as Drosophila is in developmental biology. So it's a small plant with some small flowers, and everybody's studying it. And basically, it's been very well studied. So the tip, and it's right here somewhere, of the stem consists of several layers of stem cells. And you see the structure of different layers. It's very well structured. You can actually see deep into the this piece of tissue. And tunica is just few outer layers of cells, and they're kind of all oriented kind of in that direction. Cells are very elongated. And this is another big difference between plants and say animals. Each cell sits in a domain bounded by cell walls. Okay, so this cell walls is the alternative to ECM in tissue of animals. So it's kind of maintained by cells. It's pretty stiff. So membranes of cells and microtubule layer can attach to this, and they built and rebuilt this walls. Actually, it's like living in an apartment. Okay, it's just you're sharing it with your neighbors, and you're making decisions, and you kind of share even actomycin. There are channels. But there is this stiffness, and that's clear why, because it's supposed to support its own weight, right, when it's growing, for example. So we started using cell element to account for mechanical stresses and dynamics of growth, and cells are highly polarized. But you see, if you get kind of deeper, the orientation changes. It becomes mixed, and in the middle, it's really not clear what it is. The important thing is they keep dividing, right? And there are new layers formed, and some of these cells get pushed to the side, then they differentiate. So on the sides, they differentiate into cells kind of which start new branches, or it can differentiate into cells starting a flower. And on tip of every branch of a plant, there are stem cells doing the same thing. So the big open question is how they maintain the right structure to keep growing, say, in one specific direction. And if you apply pressure, how come they start kind of outgrowing it and go back to the same direction? They're growing up, or they're growing at an angle and so on. And that's what we've been studying. And this is an apical view of a stem, or stem, this upper part of a stem. And this is the structure. So we were able to use confocal microscopy, machine learning to improve images. And we use confocal microscopy kind of to a degree, because the deeper you go, the less clarity you get with these images. So we use specific machine learning algorithm. We learn what happens at like higher layers of cells, and we go deeper. We use it to recognize kind of this deeper layers, and it works pretty well. So we have a paper on that. So basically, you see that the structure is very complex, but there is a structure. There are different layers. And what happens in upper layers is very important, as I described before. There are certain zones which can be recognized. They're given different names by plant biologists. And the regulation is very tight, because we worked with a lot of mutants, and you kind of change production of certain chemical a little bit, or mechanically perturb it, and everything goes to pieces. And by the way, getting images, Drosophila, confocal microscopy on plants, it can be kind of this technique can be applied to cells for a long time because they die, because you're using certain dye, which is kind of poison to cells. So computational simulations are very important if you want to see what happens with a piece of tissue over a long period of time. So basically, we looked at different signaling mechanisms controlling cell growth and organization. And just look at the quality of images here. And here you're actually looking at different mutants. So we're able to show that this SAM has a symmetric structure. So we looked at kind of sections, vertical sections at different angles, and we use certain metrics to compare them. They all look very similar. We use two-dimensional model, which is usually kind of something reviewers ask a lot of questions about. Why 2D is important. And so the strengths of the modeling approach we're using, we can represent mechanical properties and heterogeneous mechanical properties of cells and cell walls in this case. So depending on signaling concentrations, and we have two signals we're working with. And we have a paper in Nature Communications last year when we figured out kind of which signals are most important for SAM at certain moment in maintenance of the structure. So this is now coupled with mechanical model. So the model is very similar. It just the walls, the representation for walls, the nodes and interactions there are much stiffer. And of course, the growth is different. So the material for the walls is manufactured by cells and get transported through microtubules to the walls. So we spend a lot of time calibrating it, looking at images and some mechanical properties, so people can test mechanical properties again. And these are the equations for this system. And this is a kind of how cells look at high resolution in our model. And initially, we just looked at experimental distribution steady states for these two chemicals I was talking about, Wuschel and CK. And that's a simulation. And we were focusing on division processes. So what's very important for plant cells, because walls are so stiff, is positioning information of walls during division. It's a wide open question, what determines positioning of the wall. So I was talking about Drosophila and forming membrane during division. That's pretty clear because of cells are elongated and so on. Here, nobody knows. So we checked different mechanisms. So positioning, local positioning of the wall, you see formation of walls all the time, right? It's been speeded up. I mean, this movie, but this is a real simulation. It might be determined by chemical distributions. Chemical signaling can be determined by actually distribution of stresses. So we tried testing different mechanisms, and we can also use this model for testing hypothesis about polarization of cells. And so we looked at distribution of Wuschel, and we realized that, and that's the prediction. We recently published a paper that in apical side of the SAM, it's mostly mechanically determined positioning of walls during division, and that changes kind of the structure. But at the same time, seems it maintains the structure. And you go deeper into basal corpus, and it's actually depends to a large degree on chemical signaling. So this is a second example of application of the model. I just want to mention that early on, I was involved with James Glazer and other collaborators in developing actually cellular pods model. We switched in my group to a subcellular element model maybe 10, 15 years ago, because we started paying attention to mechanical properties of membranes, parts of membranes, subcellular elements, and the rest of it. Nothing is wrong with cellular pods model. It's easy to implement, it's stochastic. Here, we get a better connection to experimental data sometimes. Last thing I want to mention, another application of this model is to blood clot formation. And I just want to mention this. So I don't know how much you know about blood clotting, but basically when clots are formed as a result of small injury, well, next time you cut yourself, don't do this on purpose. Some blood comes out and then coagulates and covers the injury site. So prevents bleeding, right? At the same time, this clot is really essential to wound healing, and we are studying this as well. But these clots can form inside of arteries and any blood vessels in human body. There is an epidemic of pathological blood clotting. It's a leading cause of death in the United States. So people with diabetes can develop blood clots. With obesity problems, they develop blood clots in their limbs, and they get detached in the middle of the night, go to lungs, and there is nothing to be done. People die. So unfortunately, blood clotting is becoming a big issue in younger patients with COVID. So the virus, actually, I don't know how much you know about how this virus operates. It creates inflammation in epithelium anywhere in human body, especially in walls of blood vessels. They're all covered with epithelium. And as a result of it, blood coagulates. So certain types of cells, platelets, get activated and they become adhere, stick together, form small clusters. They start coagulation pathway reactions, fibrinogen is formed, so fibrin network is formed. And this small piece of clot, if it's formed, say, near the heart, and hearts get inflamed in patients, they shoot up to brain, people get paralyzed. So that's a cause of stroke in many cases, or clotting happens there. So when clot is formed, at the last moment it contracts. So after platelets are all kind of positioned themselves in the piece of this coagulated blood covering like inflamed regions, so on, they start pulling on different fibers, and the whole thing contracts and it has an appropriate structure, and it's a porous material. And don't forget, they form in blood vessels, so blood flows through the clot. And if it stops doing that, it's too stiff, it gets washed away, and as I said, with deadly consequences. So this contraction has been observed to be kind of violated or becomes pathological in patients. That's one of the main things what happens there. So using kind of blood thinners doesn't cut it, unfortunately. So anyway, we studied contraction, and we realized that what these platelets do inside of clots, they form these extensions called filapodia, and they attach them, or they attach to different fibers, fibrin fibers. That's another big component of blood clot. They start pulling on it, and they talk to each other this way. So it's like a piece of smart material. They feel that there is a, say, platelet on that, another platelet on the other side of this fiber. So they can feel the stress. They start pulling harder, and they readjust it, and they actually aggregate together. And when aggregate of platelets starts pulling, it's much more, much stronger. So they kind of share the cytoskeleton. That's like a mystery, and a lot of people are studying it. But what's really exciting also, and that's an example of new type of interaction with ECM, with this filapodia, they pull fibers underneath the platelet and they tie a knot. So you can see it right here, right? So in the end, they do it many, many times. They come up with a small rug and they sit on it and they start pulling with much harder as a result of it. So it's a new example of interaction between cells and actually extracellular matrix, because filapodia are usually they're used for sensing. They extend it, cell touches it, gets something out of it and pulls back. And it's all governed by dynamic actomyosin extension and contractility. And this is our model for the blood clot. So the model for platelets is simplified, computationally very heavy. Just kind of look at different zones and different strengths. We're training different strengths and different number of filapodia for individual platelets. And we have a model for the 3D model for a fibrin network, and can bend, extend, and the rest of it. That's like representation of real simulation at very high resolution. And this is a simulation. You see it's contracting. The other discovery was the fact that they densify the fibrin network. So cells are known to use filapodia for sensing. Cancerous cells use filapodia to move through the extracellular matrix. They don't remodel it. Well, they remodel it sometimes, but it's kind of very drastic. Here, gradually, they densify ECM close to this platelets, and that's the way they actually aggregate. And we looked at this densification at high resolution and kind of removed some of the visual data just to let you see where this accumulation happens of fibrinogen, the white stuff on the surface of platelets. And then we also classified bundles. So as a result of contraction, bundles of fibrin threads are formed, and you're looking at the cytoskeleton of contracting clot. So once again, it's like a third example of interesting type, very different type of interaction between cells and extracellular matrix. And I think I stop here. Just want to mention my collaborators on the Drosophila project. It's Jeremy Zartman, it's his lab. And then Ali is a former postdoc. He's at Cornell now. And Wei Tao Chen is my collaborator, faculty member at UCR. Kevin Tsai is a postdoc. Nilay Kumar, graduate student, and Jennifer and Alicia are graduate students, and Ali is a physics student. And it's funded by Modulus grant from NSF. And the plant project is funded by NSF as well. It's a program between DMS, NSF, and IGMS, NIH. And Dr. Gohal is a biologist, his lab, and Kerrit Rodriguez is a postdoc. And Amit Roy Chaudhuri is a faculty member in image analysis, electrical engineering. This is his student, Calvin. And just want to invite everybody to visit UCR as soon as possible. This is our new building. We have a center on quantitative modeling in biology on the third floor of this building. And these are like pictures of our neighborhood. And thank you for your attention.
Thank you, Mark. This is great. My words were not perfect before, but I hope everybody could see how your jumping systems, there's different models, there's different species. I hope the community can appreciate this breadth. I have a question, if that's okay, I can start. To be honest, your wing model remind me of the brain. I've been studying the brain. So if you're interested in a new system, what I've learned is that the bottom cells of the brain, they actually go up and down too, and only divide in one locations. And so it makes me wonder or even think about maybe you can help explain the folding in the brain, because I know there's a lot of like studies in that area, and I wonder how much we can push it to apply to different also species, because like the brain of the mouse has different layers. So I think it's just 3 layers, where the human brain has more layers. And if the Drosophila only has 2, I wonder if that makes difference. So this is just more of a thought question.
So folding obviously is very important biological phenomenon, right? So we're looking mostly on mechanical side of it, and it is mechanical, but there is a signaling side. It's very important to look at what kind of signaling is involved. So in Drosophila, that's been studied, DPP and other components, and the same for SEM. But this upper part of, if signaling is known for brain, or people have some hypothesis how it kind of controls division rates, for example, right? Or polarization of cells, or redistribution, actomycin activity. I was trying to put emphasis on kind of subcell processes in my talk. So when I talk about, say, philopodia forming, its cell is changing its shape in a very dramatic way. Yeah, it's all controlled by signaling.
Well, I thought it would just be cool to push, right? Like, so we can't officially like study the human brain unless it's an organoid, but I just wondered that if you know something about the wing, and then we can then push it a little bit further to, let's say, the mouse, and then maybe then we can extrapolate to other species that may have multiple layers. So I don't know if we need to know, or more maybe tell others what you might think is happening. To me, that would be quite fascinating.
Right. So first of all, the hope for my talk is to publicize subcell element. When applying it, there are a few other groups doing the same. Just we're very willing to share the model and work with other groups on kind of applying this modeling approach. Because, say, Cellopods model has been very well publicized, CompuCell 3D group, and they're doing great things. So we're in the initial stages of publicizing this subcell element approach.
Leah had to go. Sorry.
Leah left a message on.
Yeah, she said she'll reach out.
Yeah.
Does anybody else have questions?
Yeah, are there other examples of what you showed very clearly, the blood clot is used in wound healing, and at the same time, this is a pro side of it, and there is a con side of it in stroke. Are other examples of things that are bad, which can be also good in the same vein?
So the point is, of course, the whole idea of studying, say, Drosophila development is to deal with actual birth defects in human embryos. So studying pathology or studying what can go wrong in the early stages of embryo development can be used to produce new drugs to deal with, or new strategies to deal with birth defects. At the same time with, say, plants, studying kind of sensitivity of this mechanism of maintaining structure of SEM, or part of a cell, is very important. Kind of then new plants hopefully can be engineered. A lot of plants are disappearing from California, right? I'm sure from other places we're having drought and everything else. So kind of engineering new types of plants which would be kind of more resilient to changing conditions, that would be the goal of that kind of study. And I would say the SEM is the most sensitive part of a plant. It's not going to grow if something goes wrong with that. And on blood clotting, of course, if you figure out what's wrong with this kind of, say, contraction behavior or blood clot, then a new drug can be developed to deal with it. So at this point, the main drugs used by doctors, patients with this kind of developing this condition as a part of COVID response, just blood thinners. But what was shown recently that this clots in brain mostly consists of fibrin network. And so most drugs, blood thinners, kind of impact adhesivity of platelets instead of fibrin network. So the whole industry, pharmaceutical industry, is kind of refocusing on different type of component in the blood clot. So you need to target fibrin instead of adhesivity of platelets.
That's great. I have a technical question, and this is about the shape, the hexagonal shape that appears also in a solid state and many other phenomena. And the question here is, can it be justified as optimal here? And also, how is it connected to the curvature and curvature forces?
Okay, very good question. And of course, we'll look at physics data as well, like liquid crystals modeling and the rest of it. So the shapes you see, these are steady states of the system. So these are equilibrium shapes. Of course, if you look at it from a physics point of view, from a biological point of view, cells control the environment. So after division happens, or division is happening, it's not a physical process at all. So after they divide completely, and that might be impacted by distribution of stresses, it goes out of equilibrium. Biological systems are out of equilibrium. That's the main difference between physics and biology. But after division, this kind of big events happen, everything goes to equilibrium state again. There's a distribution of stresses, and this is how we treat it in a multi-scale model. So division is division. We don't look at, say, minimization of energy, so on. We're just trying to mimic what happens in experiments. But after they divide, we give them time to go to equilibrium, relax to equilibrium. So this is a soft tissue, of course, but then it's a soft matter physics, but at the same time, it's controlled by cells signaling, and that makes it biological.
Thank you.
Mhm.
Nikhil has a question in the chat. Platelet interaction seems to have been modeled mechanistically or deterministically. Does it make more sense to model it stochastically?
So actually, adhesivity, when they get close to each other, we have a kinetic model for that. So there's bonds which are formed, for that we use Monte Carlo. Not in every simulation, but we developed that kind of approach as well. Just think about how long it takes to run this simulation. So we actually been parallelizing them, the simulations on GPU, very large GPU clusters. So we're using very large cluster at La Jolla, like a national supercomputer facility. Definitely, when you start looking at two cells adhering with each other, or you look at filopodia attaching to a fiber, there's a molecular level events and bones, and you need to use Monte Carlo for that.